Hello, my name is Carly Whitlock, and these are my partners, Hallie Saucier and Lauren Rhodes. We are DNA analysts.
It is our job to sequence DNA from crime scenes to create DNA profiles. We take DNA samples and use processes such as the polymerase chain reaction and gel electrophoresis to create DNA fingerprints. We use this information to identify suspects in crimes and use it as proof in court cases.
Gel electrophoresis is a step-by-step process that requires patience and technique to get the best results. Each step in the process has possibilities for human errors to occur, which in the end would result in wrong information and wasted time. In addition, gel electrophoresis is an additional step taken by DNA analysts in the lab, so it also causes less of a convenience to accuse a suspect at the crime scene right away.
We are here to introduce our product, the DNA Severance scanner. This mechanism is used at crime scenes and is used after the extraction of DNA from a victim or victims.
The DNA sample is put into a dish and inserted into the mechanism. It is then separated, analyzed, and converted into a code that will be stored in the mechanism's database.
The machine is made of steel with the same wiring as a phone or computer for the capability to access worldwide databases. It enables us to scan DNA without going through long processes. It prevents cross-contamination because the wiring allows samples to be sanitized immediately. So let's say that we're at a crime scene. There's a victim killed, and our job is to collect DNA samples. Luckily, a hair was found on the victim's body, and the hair does not belong to the victim.
The piece of hair is a perfect sample to use for this crime investigation because there are possible skin cells on the hair that could cause cross-contamination that could produce different results. Typically, DNA analysts would take the sample back to the lab and separate the DNA. The process takes a large amount of time, and results can be inaccurate because of human errors.
At crime scenes, DNA analysts work together to get the sample of DNA from the victim or victims for further testing at the lab. By saying this, the analysts would probably have an easier time retaining the DNA information using the DNA Severance scanner, due to the availability and convenience of the mechanism itself.
Oh, hello there. However, the DNA Severance scanner makes processes shorter. Here we have me, a DNA analyst, collecting the sample, a piece of hair. Instead of going to a lab, I put this DNA into the Severance scanner. This scanner will isolate and separate different DNA that has been cross-contaminated. DNA is then scanned and put into a code. This code automatically is stored into a database that can be accessed with any computer. This replaces other processes and creates an easier storage for information. There are many advantages that come with our product, the DNA Severance scanner. For example, it eliminates the need for physical and complicated processes in a laboratory after a crime scene investigation. It also eliminates the process of gel electrophoresis that takes a lot of time.
This saves a lot of time and money compared to using machines and gel electrophoresis. It prevents DNA from undergoing cross-contamination, and this will prevent mistakes and errors that will result in wrongful accusations.
However, some disadvantages can occur while using our product because malfunctions can occur due to technology use, as it would with any new piece of technology. Also, programming mistakes can lead to inaccurate results for information.
And of course, there's human error that could change the outcome. If DNA is mixed with the DNA samples, then the DNA would be recorded without knowledge of the error. Altogether, these things could make this product less trustworthy.
Similar to other products, there will be production errors, but with the financial help of experienced entrepreneurs, we can receive professional advice and guidance. We are looking into designing personalized training programs for our customers and those who endorse us. This will be very effective because it will ensure that our customers receive correct information and instructions on how to use the product.
Training for customers, we are asking for $5,000, and materials, we are asking for $10,000. Along with that, we are asking for $30,000 for research and testing for our product. And we are asking for $20,000 for the production of our product. In total, we are asking for $65,000 to develop and create our product. We are looking to keep our company small scale until the technology develops and becomes more effective.
And don't forget, remember to vote for us on Splice Tank.